Dear learners, welcome to this video. The topic for today's discussion is based on phase contrast microscopy. Now, during the last lectures, we had discussed a lot about the different versions, the different types of microscopy, the features, and the different light properties which govern the functioning of a light microscope. And in today's topic, we shall be discussing about the phase contrast microscope. Now, till now, the different versions of microscopy and the features, they all gave us a different perspective. The different technical modifications which could be done within a microscope and the features which are associated and so on. So let us see the learning objectives for today's topic. The learning objectives are, first is, we'll try to describe the basic concept of a contrast and to elaborate on the principle and working of a phase contrast microscope. So let us see the basic principle and working of the phase contrast microscope. This particular technical modification in the microscope, it converts light differences in the refractive index and the cell density into the easily detected variations in light intensity, and it is an excellent way to observe the living cells. Now here there is a technical modification made, and that too at the level of a condenser. It has an annular stop, which is an opaque disc with a thin transparent ring, which will produce a hollow cone of light, which we shall see now using a diagram. So once this cone is passing through the cell, some of the light rays are bent due to variations in the density and the refractive index within the specimen, and they will be retarded by about 1/4 of a wavelength. So this is one point to be noted, that when there is a light beam passing, and now because of the technical modification which is done at the condenser, it has an annular stop. So it is an opaque disc with a transparent ring. So this will produce a hollow cone of light, and when this cone is passing through a cell, some of them will be bent. Now the deviated light is focused to form an image of the object, and those which are not deviated, they will strike a phase ring in the phase plate. It is a special optical disc which is located in the objective. So this is one more technical modification. While the deviated rays will miss the ring and they will pass through the rest of the plate. So if the phase ring is constructed in such a way that the light which is not deviated passing through it is advanced by 1/4 of a wavelength, the deviated and the undeviated waves will be about 1/2, half of a wavelength, which will be out of phase and will cancel each other when they come together to form an image. So here the background which will be formed by the light which is not deviated will be bright, while the particular object which is not stained, it will basically appear dark and well defined. So this type of microscopy is called as the dark phase contrast microscopy, and color filters, they are often used to improve the basic image. So dear learners, for this particular topic, the reference book could be Microbiology, 5th edition by Prescott. Let us see the diagram now, the working picture. So this particular diagram shows the basic properties and the light optics of the dark phase microscope. So first we'll see from the bottom what type of modifications are made. So there is a annular stop which you can see in the diagram. So the direct light rays are advanced 1/4 wavelength as they pass through the phase ring. So you can see direct light rays are advanced 1/4 wavelength as they pass through the phase ring. So most of the deviated and the diffracted rays of light will pass through the phase plate, which is unchanged because they miss the phase ring. And the diffracted rays are radiated, they are basically retarded 1/4 wavelength, which will be after passing through the objects. So here the condenser has this modification which is an opaque disc. So this is where the technical modification is. So here you can see the phase plate. It has a phase ring. So the amplitude, which is contrast, is produced by the light rays that are in reverse phase. So the dark image will be observed with a bright background. Let's move on further and see the mechanism by which the contrast is generated using a phase contrast. So if you see, this is a bacterial cell. The ray which will be deviated by specimen is 1/4 wavelength, which is not in phase, it is out of phase. So here we have a phase plate, just we saw within the diagram. And the deviated ray is half of the wavelength, which is out of phase. And now we have a lens. So now both of rays, they will now come at one point. So the deviated and the rays which are not deviated, they will cancel each other out at this point. So here is where the basic type of contrast is created. These are some of the pictures I have tried to show you how the particular bacterial specimens will look like. In the first picture we have Spirillum volutans. It's a very large bacterium with some sort of flagellar bundles, phase contrast microscope. The second is for the Clostridium botulinum, responsible for botulism with the subterminal oval endospores. The lower one is Paramecium, stained to show a large central macronucleus with a small spherical image. So this is how I just wanted to show you the different types of specimens and how they could be observed. So thank you for listening and keep exploring the world of mini wonders.